Hello and welcome back. Today we will focus on depression. There has been a long-standing debate in the field of psychiatry about subtypes of depression. And the DSM in its various incarnations through DSM-3, 4, and 5 has been attempting to what is called carving nature at its joints. And it has been spectacularly unsuccessful, as evidenced by the fact that we cannot really find genetic markers to correlate with major depression, as opposed to, for example, schizophrenia, where reliable markers in genome-wide association studies have been found. And the reason supposedly is that we don't classify depression correctly. So now here's a paper that just appeared in Nature Medicine in January of 2017, in keeping with our philosophy of getting you the latest and hottest in psychiatry and neuroscience. Now, interestingly, this paper appeared in Nature Medicine rather than Molecular Psychiatry or one of the other journals that focus on psychiatry. And I would be curious about the editorial history of this paper, whether it was submitted originally to Nature Medicine or whether it got stranded in some of the other journals because of being rejected given its approach. So briefly then, what this paper is trying to do is classify depression without any preconceived bias. So rather than imposing the diagnosis from the outside, this paper attempts to use markers derived from brain activity in an unbiased, computerized search using machine intelligence to identify possible subtypes of depression. And this is the basic strategy. So an initial discovery sample was used, and subsequently it was optimized and validated in a large number of patients, 711. And not only that, but a replication study was then launched for another 477 patients, which was able to separate controls from depression and identify 4 subtypes of depression. Furthermore, the authors then went ahead and showed that one of the subtypes responded particularly well to transcranial magnetic stimulation, whereas other subtypes did not. Now, to put the icing on the cake, the authors also showed that the diagnosis derived from computerized approaches did not respect the boundaries imposed by our diagnostic categories, which are fond of separating anxiety and depression. So eventually they showed that generalized anxiety disorder has significant overlap with depression. Now this should be a no-brainer because we all know that in clinical psychiatry we use serotonin uptake blockers both for the treatment of depression as well as for the treatment of anxiety. Now, to show specificity, they were able to identify that schizophrenic patients were clearly not identified by the algorithms and by the machine learning approach, as shown here in part C of this slide. So here then is the idea to use machine learning. When you do massive correlation between voxels in the brain with each other to identify connectivity patterns in the brain, you wind up with a huge number of matrices and data points that are impossible for a human to look through and to find clusters or patterns. So machine learning approaches were used, and you may have heard about machine learning. It's increasingly being utilized in imaging studies, in genetic studies, and other studies that deal with a large amount of data sets that are difficult to analyze in any other way, but with machine learning, in order to find patterns or clusters. And I show you a paper here that appeared in Science magazine, and which is included in your reference set, and you can look at the PDF online. So this is a paper on machine learning trends, perspective, and prospects, and it covers a number of things such as visual recognition, for example. It also covers the idea of classifying visual patterns and, for example, differentiate oranges from lemons or starfish from birds. So in visual recognition, such as used by Google to identify your particular face among thousands of other faces, these are done by machine learning algorithms. Here you see a basic style of approach here. It involves inputs and then convolutional feature extractions. We will not get into this at this time. It's been also used in studies in genetics to identify which genes are somehow connected with each other in metabolic trees. Finally, here is a broad overview as to where these technologies may go, such as self-driving cars, and they may come to your iPhone before too long. So let's get back to our main point, which is depression. So what these authors then are finding is that there are different biotypes of depression. Now, all biotypes, of which there are 4, according to the study, include a common core of altered connectivity involving prefrontal, orbitofrontal, posterior cingulate, and parahippocampal cortices, as well as nucleus accumbens and pallidum, which is part of the striatum. Now, biotypes 1 and 2 score highest on anergia and fatigue as compared to healthy control, and they show reduced connectivity with anterior cingulate and orbitofrontal cortex, whereas biotypes 3 and 4 are high in anhedonia and psychomotor slowing and show increased thalamic and frontal striatal connectivity. Now, as the icing on the cake, biotypes 1 and 4 also score high in anxiety and have an abnormal frontoamygdala connectivity, as you would expect, because we all know that frontal cortices have the job of attenuating anxiety signals originating in the amygdala. So that's quite an amazing result, given the fact that this was done in an unbiased machine learning kind of a way. So here are some of the major approaches and results. You can see that there is a time series here of the bold signal, the wiggly lines here at the bottom of the slide, and you can show various cortical areas that show unusual connectivity. This is a so-called connectivity matrix. You just correlate the various brain areas with each other, and you can see that there's a diagonal going through, which is the autocorrelation. So default mode network versus default mode network at the lower side of the slide, of course, shows a very high correlation in hot colors. The same is true for PFTC, this is the prefrontal cortices, and so on and so forth. But you can see that other areas do not correlate as well. So here is a correlation which shows the abnormal anhedonia-related connectivity and the anhedonia clinical scores. And you see there is a very strong correlation which demonstrates that the connectivity ratings and the clinical ratings of anhedonia are extremely tightly matched. So what we have here really is an implementation of the idea not to look for categories that are imposed from the outside by a set of criteria, but rather following the research diagnostic criteria proposed by the NIMH not too long ago, to look at symptoms that make sense in terms of brain circuitry and identify those as driving the disease or being at the core of the disease process. Same is true for the anxiety clinical ratings and the anxiety-related connectivity scores I showed you in a previous slide. Again, a very tight correlation is found, and you can see the circuitry at the bottom of the slide here for your review. So this then is the cluster analysis, the hierarchical clustering analysis, which shows you the distance between the different biotypes in a mathematical kind of a way. And what we find here is 4 clusters that the authors then determine are the 4 biotypes. So we arrive at this by a strictly mathematical approach, using data from resting state brain activity from fMRI, and then subject this data to a canonical correlation analysis and a pattern recognition approach that uses machine learning. So here you can see how these clusters separate from each other. You have anxiety-related scores on the y-axis and anhedonia connectivity scores on the x-axis, and you can see the clusters separate quite neatly from each other. So there is an anhedonia-related subtype of depression. There is an anxiety-related subtype of depression, and these can be separated very reliably by just looking at fMRI data. Now, back to the common neuroanatomical core that is shared by all depression. And that involves, of course, some regions of interest here that are shared and encompass frontal lobe activity, the posterior cingulate cortex, and other regions of the brain. Now, of the 17 symptoms that are identified by the Hamilton Depression Scale, 3 were present in almost all patients with depression, namely changes in mood, sadness, hopelessness, and helplessness, anhedonia, and anergia or fatigue. Those seem to be shared in all depression. And then all hell breaks loose, and the data separate into 4 different categories. So here then is the heat map of the pattern of abnormal connectivity shared by all the 4 subtypes. So we will not go into this in detail. I refer you to the article by Drysdale and others. Now, across all the subtypes of depression, abnormal connectivity is found in a core identified by principal component analysis: depressed mood, anhedonia, and anergia. You can see there is a very strong correlation between the abnormal connectivity and the clinical rating z-scores. Now here we have those things that stick out, that don't fit and constitute the 4 biotypes. So superimposed on the shared biological core are distinct patterns of abnormal functional connectivity that differentiates these 4 biotypes from each other. The size of the bubble here is a statistical measure of separateness from the other biotypes. And here's the reduced connectivity in frontoamygdala networks in the biotypes. And you can see that it's only in some of the biotypes here that increased anxiety correlates with abnormal connectivity, namely in biotypes 1 and 4. So this then makes the case that anxiety and depression may be two sides of the same coin, heads and tails, that you can flip over, but not for all depression. Some depressions run more purely and exclusively with depression symptom clusters. That is important to note because the responsiveness to treatment may vary according to which biotype you belong to. So here's another way to break down the biotypes and how their symptoms cluster. For example, biotype number 3 here in a magenta type color scores high in anhedonia, psychomotor retardation, whereas biotype number 1 is low in anhedonia but much higher in anxiety, as an example. On the right-hand side of the slide, you can see a plot that shows the separation of the biotypes by combining all the statistical measure into one score. Now, so this is the transcending of diagnostic boundaries. These data don't respect the data that come from the DSM-3, 4, or 5, because as you can see, there is significant overlap here of depression and anxiety, with a lot of shared, 546 shared abnormal connectivity features. So this is a way then to separate out different types of depression, which then might be more fruitfully related to genetic analysis in the attempt to identify genetic markers. So although none of the patients with GAD in this analysis met clinical criteria for the diagnosis of depression, more than half nevertheless were classified by the machine learning algorithm as belonging to one of the depression biotypes. And the majority of these were assigned to the anxiety-associated biotype 4. So you may have depression or a depression signature, biological signature, brain signature, present in your patient, but it's not really apparent and you may totally overlook this type because the diagnostic criteria will not allow you to make this determination. Now, in terms of the heritability of depression, analyses have been trying to identify genome-wide association markers for depression and have been largely unsuccessful. So recent initiatives to rethink our system of psychiatric disorders in investigating their neurophysiological and genetic basis begins by stratifying subjects into subgroups shared by neurobiological substrates. And here's some references that you can click on and look at the original papers. Dr. Insel in particular, who used to be the leader of the NIMH, makes the case that brain disorders, precisely. So we are now are able to map a psychiatric diagnosis into brain circuits, and that should be our goal. There is a paper here by Chaduri. This is a optogenetic study that shows depression-related behaviors originating in midbrain dopaminergic neurons in nature. I highly recommend this to you. A pathfinding study using optogenetics to clearly demonstrate, not by correlation, but by the proof of causation of depression-like behaviors in rodents originating in midbrain dopamine systems. Now, this is not the first paper that has been doing this general approach. There was a paper some time ago using this strategy in identifying subtypes of psychoses using brain-based biomarkers. And here it is. So this paper appeared in the American Journal of Psychiatry in 2016, and it used a variety of markers such as eye tracking, saccadic movements, responses to auditory stimuli, and I want to show you some of the correlations here, and you can see that a saccadic error, a score, differentiated biotypes in schizophrenia, as well as evoked potentials, and in relatives this could be demonstrated as well, which I will show you in a slide. So here the test, neuropsychological, stop signal, saccadic, controlled, and auditory evoked potential. And a multivariate taxometric analysis identified 3 neurobiological distinct psychosis biotypes that also did not respect clinical diagnosis boundaries. This highlights a possible advantage of neurobiological versus clinical categorization schemes for differentiating psychotic disorders. So here you go. You have the normal controls here in the middle, and you have biotype 1, 2, and 3 clearly differentiated by these different markers. And you can see that the relatives in the left side of the slide also showed a similar signal. This was also seen for sensory motor reactivity. So this did not involve going into the data from fMRI, but rather in making educated guesses about what type of neuropsychological abnormalities might be present in psychosis, and use those to classify patients rather than clinical diagnoses. And as you can see, you can successfully identify subtypes of psychosis by focusing on subtypes of neuropsychological dysfunction rather than an overall global diagnosis. Here's a number of references that highlight the various approaches that have been taken. All of these are clickable if you go to the presentation page in our website. You can click on each one of these and look at the original PDF file. Again, the strategy is to define depression subtypes by clustering subjects according to distinct whole brain patterns of abnormal functional connectivity in resting state networks, unbiased by assumptions about the involvement of any particular brain region, and tested in a large multi-site data set. Now let's focus on one of these previous examples to subtype depression, which was really quite prescient. It's the linking of the HTLPR polymorphism, the serotonin transporter gene, with the interaction of the cingulate and the amygdala, identifying a genetic susceptibility for depression. Now here's just the anatomical part of the study, namely the cortical density in the subgenural cortex, the area, Brodmann area 32, roughly. We have talked about this area before in our talk on depression entitled A Walk in the Green. The area here lit up in red is that area that cools down when you take a relaxing walk in a green environment and experience a decrease in the score for rumination. This seems to be a critical area for depression. It's also the target in many deep brain stimulation approaches to the therapy of treatment-resistant depression. And here you can see that you can separate the genotype by carrier, by the full expressed genotype, and you can see that the signal from brain imaging are separated according to which genetic subtype you belong to. So functional connectivity abnormality from cingulate to the amygdala is highly expressed in only one particular genetic type. One more demonstration of the power of separating depression by subtypes and predicting treatment response. So this is anhedonia, and the reward circuit connectivity is able to distinguish responders from non-responders to repetitive transcranial magnetic stimulation. Our target paper, which we began this talk with, also showed this and demonstrated that the left dorsofrontal cortex stimulation only works in patients with one or two of the biotypes that these authors identified. So the treatment response then was strongly bimodal, with individual patients showing rather minimal or marked improvement. Compared with responders, non-responders showed markedly higher baseline anhedonia symptomatology, including pessimism, loss of pleasure, loss of interest in previously enjoyed activities. Non-responders showed significantly lower connectivity through a classical reward pathway comprising ventral tegmental area, striatum, and region in the ventromedial prefrontal cortex. So this is the setup. You can see the magnet applied to the head of this young lady. In part B of the slide, you see a little marker that shows where the magnetic coil is aiming at, namely the left prefrontal cortex, and you then apply the stimulation to the scalp. This is the area of the brain that separates responders from non-responders by using a statistical measure called betweenness centrality. So that's the area that most clearly separates the two groups from each other, namely responders and non-responders. And the paper makes reference to a paper by Dieckhoff, The Role of Human Ventral Striatum in the Representation of Reward Magnitude in panel B. And you can see that this subgeneral area is part of this particular circuit. Now, here you see the difference between responders and non-responders on the left side of the slide, and it's a rather dramatic difference. It doesn't get any better in biological psychiatry than this clear separation, predicted by brain imaging studies. Now, on the right hand of the slide, you see the correlation of the HAM-D improvement score and the normalized anhedonia composite score. So the more anhedonia you have here, the more the improved the HAM-D is decreasing. A very striking correlation. So this makes sense both in terms of what we know about the reward system, and it puts a highlight on the power of brain imaging studies to predict the response to treatment. So here's a paper on neuroimaging treatment selection biomarkers for major depression. This came out quite some time ago in JAMA Psychiatry, making the point that you can differentiate the responders for cognitive behavioral therapy in panel B and escitalopram according to the activity of the right anterior insula. Now, the insula, as you know, integrates signals coming from your body, kind of your internal milieu. And you can see a clear differentiation here between CBT non-responders and CBT remitters in panel A of the slide, and also a separation of escitalopram remitters and escitalopram non-responders as stratified by the activity of the right anterior insula. Again, this was proposed as a biomarker predicting treatment response in depression. This is critically important because we treat all depressions alike. Everybody gets an SSRI, and you can see here that just from this slide, you only expect maybe half of the patient to be appropriate patients for an SSRI because the rest of your patients have a different connectivity problem that is not amenable to treatment with your proposed drug. Now, none of this ever made it into the DSM-5, although the DSM folks have long talked about updating their approach to include neurobiological markers. It hasn't happened yet. And this is yet another finding here, combined with the key paper at the beginning of this talk, indicating that it's high time for us to include neurobiological markers in order to maximize treatment response. So here I have a number of slides that you can click on. This will pop up a large number of papers that you might want to peruse, all of which go into increased depth in discussing these issues and making the case of biomarkers and a new approach in psychiatry looking at brain circuitry as the starting point for classifying illness rather than as an endpoint, and getting away from the DSM-5 classification, which has neither been helpful in identifying genetic markers nor has it been helpful in aiding our treatment strategies. So this is just one major item to show you we need to rethink psychiatry in a more global way. We will have more talks like this coming up, particularly in subdividing psychosis, and making the point that we need to form models that go from the genes to cells to circuits to symptom manifestation, allowing us to then intelligently select appropriate treatment responses. Thank you for your attention, and we'll see you soon again in a presentation here from Behavioral Health 2000. And Happy New Year to you, and all the best for 2017. Thanks. Bye.